and we'll try and recap. I mean, I've shared the worksheet. We'll do the worksheet later on, and I've shared the notes as well. I said, but before that, we had to finish this. We were almost at the end of transition metals, and I had talked about monodentate ligands. Okay, these were ligands that were able to form just one coordinate bond. And then we talked about bidentate ligands, and there were two bidentate ligands. You had ethane dioate ions, so they've got lone pairs. Two sides of the molecule has lone pairs, and then there was ethane 1,2 diamine. So this one, I showed you how the bidentate ligand was actually forming dative bonds. Okay, that was right in front of you. So, is this clear, Isaac? Qasim, is this clear what a bidentate ligand is?
Yes.
So now there's another one which is, so it's going to be a molecule that can form two dative bonds. And then there's a polydentate ligand. The polydentate ligand can form many dative bonds. Dentate means teeth. So that is basically lone pairs. So it's a species that can form many coordinate bonds or dative bonds with the metal cation in a metal complex. So there's one polydentate ligand that you should know of. You should actually just know the name of that molecule. Drawing the molecule or remembering the molecule, that's kind of difficult. Just one second, EDTA ligand. I need to draw this. So I just want to see one thing. How does the molecule look like? So this is so even I don't remember what an EDTA looks like, but that's a poly. I'm just going to show you. No one is going to ask you to draw this. So this one is CH2, and there are actually two CH2s in the middle. There's an N with its lone pairs. There's another N with its lone pairs. And then it's got two branches. The N is making 3 bonds. And there is CH2. And then there is C double bond O minus 1. So this side has another lone pair. Then you have CH2. And then you have C double bond O and O minus 1. So there's another lone pair over here. So on this side, the N is making 2 more bonds. And it has the same thing. So it's got CH2. And then it's got C double bond O and O minus 1 with lone pairs. And then there is CH2. Let me just check whether there is CH2. Yes. And then there is C double bond O and O minus 1. And that has a lone pair as well. So this is what EDTA looks like. I mean, you should be able to identify it. No one is going to ask you to draw this. It's EDTA, EDTA. And it's going to have a charge of -4. So that's what a polydentate ligand looks like. I'm going to highlight the parts of the molecule that will be, I mean, this one would form a dative bond. This lone pair over here would form a dative bond. This lone pair over here would form a dative bond. This lone pair would form a dative bond. So they're basically the whole entire molecule has a total of 6 lone pairs, and all those lone pairs are capable of forming dative bonds. And so just the knowledge, because maybe they might ask you to draw this, but they've never actually asked you to draw this. It's part of your syllabus. And I guess the only reason they've never actually asked you to ever draw this is it's pretty hard to remember what the EDTA ligand looks like. Furthermore, we're going to try and see how it's going to form multiple dative bonds, how one molecule is capable of forming multiple dative bonds. So there are, let's say this is my x-axis, this over here is my y-axis, and then I have my z-axis. So I'm going to just try and attempt how this is going to happen. So that big molecule is going to wrap around this metal line, whatever that metal line is. Let's say that metal line has a +2 charge. So this entire big molecule, for example, the N lone pair over here is, let me choose another color. Okay, that N lone pair would try and form a dative bond with the metal line over here. And the oxygen, which was -1, was going to bond with this. And if I try and, there's probably going to be a double bond over here. So the section that I've drawn is, at the moment, the section that I've drawn is, it's this one. It's the N lone pair. And then there's O-1 lone pair, and then there is CH2 and serial bond O in the middle. So this is your CH2, and this is your serial bond O in the middle. And then that N is connected to the other one. So right below that, there's going to be another O-1, and it's going to have its lone pairs. And that N is, I'm going to just copy this quickly. So I'm attaching this thing over here. There are two CH2s in the middle. So the two CH2s and the Skeletal formula would be like this. And after that, there's O-1 and its lone pairs, and there's going to be double bond O, and there's going to be a CH2 that's going to connect with it. And then I'm left with another CH2. So this goes and this N at the bottom is also going to lone pairs should be on this side. So anyways, if you can see the board, what is happening is, I mean, it's the entire molecule over here that is wrapping around this metal line. It's completely wrapping around this metal line. There's this serial bond O-1. It attaches. The serial bond O is connected to this N. This N over here is then connected to the other N. So there's CH2, and then there's this other N. This other N is connected to two carboxylate groups. So there's serial bond O-1, and then there's this serial bond O-1. The O-1 lone pairs are attached to or forming a coordinate bond with the metal line, and so on. So is this clear what a polydentate ligand looks like? EK, is this clear? Umar, Qasim, Ishaq, is this clear?
Yes.
EK, so remember, you had a bidentate ligand, and remember, there were only two bidentate ligands that are in your syllabus. There's this one and there's this one. That's it. and you just have to draw them. The two bidentate ligands would be connected to the metal ion. And there's only one polydentate ligand whose drawing they've never actually given in the exam. They've never actually asked you to draw this, but they can. But it would still be a very, very impossible thing to draw this. Okay, but you just need to know the idea what the ETA molecule looks like. So there's, if I write down the complex, it's M+2, and then there is ETA, which is -4. So in total, overall, the charge on the complex +2 and -4, that's going to be -2. That is going to be the complex that's going to be formed when ETA reacts. And this thing is very common. Polydentate ligands are very common in complex biological molecules. Are common. So they are very common in complex biological molecules. What happens is that when you have a like iron in your blood, the heme group is an example of a polydentate. I think there are 4 heme groups in a hemoglobin molecule. I think there are 4. I'm not sure. TK, no one is going to ask you. That's biology. But the heme group kind of looks like it's a complex molecule, except that there's an iron that is stuck in the middle that's kind of forming a complex. It's something like this. It's this thing is, I mean, remember, not in syllabus. They can ask you. You just need to remember the drawing is not important. The concept of a polydentate ligand is important. So, for example, I'm going to show you how the hemoglobin molecule works. It's basically an iron ion, which is normally +2. And that +2 iron ion is stuck, natively bonded to lone pairs that are coming from N, from all sides. So it's stuck with that. And that N, obviously, I mean, they are all, I mean, they're part of a much, much complex molecule. So the bonds, they just continue. And it just, I mean, I'm just going to draw a rough example that their bonding just continues, but there is an iron ion that's stuck in the middle, forming a complex. So your red blood cells have this in them. And how does the oxygen get attached to it? The O2 molecule has lone pairs. So there's one side of iron ion which is open. This oxygen molecule gets attracted to this iron ion which is stuck in the middle of the heme group, and it datively bonds to it. So oxygen molecules, lone pairs, they datively attach to iron 2 ions in the heme group. So the point I'm saying is the drawing is not important, the concept is important. So the way they're going to ask the question is, I mean, they might draw the drawing for you, and they'll just ask you, how does the oxygen molecule gets absorbed onto the iron ion? So you will say that it's forming a coordinate rotative bond. It's not going to be really complicated. It's just going to be a 1-mark question, a small 1-mark question. The only thing you need to know is you just have to you got to have an idea of what a polydentate ligand is. Okay, ये clear है? Is this clear? About polydentate. Is all clear? Umar, Qasim, is this clear?
Yes.
I said now, so that's it. You just need to know monodentate, bidentate, and then there's just the concept of what a polydentate is. We're gonna come to what's left? Stereochemistry. Stereochemistry is basically stereoisomers. In metal complexes. It's the same, almost the same concept as in organic chemistry. When complexes are getting formed, they tend to form different types of isomers. In organic chemistry, you studied two types of stereoisomers. You studied geometric isomers, which were your cis-trans. So these were your cis-trans isomers. ठीक है, अब यहाँ पे ये कि when you're dealing with cis-trans isomers, they are different types of complex. We basically studied two types of complexes. The first one was octahedral complex. So for example, I've got Cu, which is surrounded by 4 NH3s, and there are 2 water molecules. And this thing has a charge of +2. Geometric isomers or cis-trans were that the same type of groups can be on one side, or the same type of groups could be on opposite sides. So let's quickly draw this as copper +2 stuck in the middle. And you have your octahedral complex. Okay, so that's your X, Y, and Z axis. Now, 4 ammonias and 2 water molecules. What can happen is that the water molecules, they could be attached over here, and the ammonia molecules could be attached on this side. Plus, the entire thing is, so the entire thing is going to have a charge of +2. So that's one type of complex, an octahedral complex. 4 ammonias and it's got 2 water molecules datively attached to it. Now, if you look carefully, the water molecules are attached on opposite sides. If you look at the molecule. Now, there could be another version, another arrangement where the ammonia would probably be attached. The ammonia is going to be attached on the top, while the water could be attached on this side. Now, if you look at this molecule, the water ligands are attached on adjacent sides. So that is what geometric isomers is. That arranging 4 ammonia and 2 water molecules around the copper ions, you've got 2 different arrangements. Either they're going to be on opposite sides, or they're going to be. So this one is opposite sides. And this one is on the same side. Same side is cis. So that's your cis arrangement. Opposite side is trans. Okay, so is the concept clear about how stereochemistry or geometric isomerism could arise in octahedral complex? You clear? Qasim and Umar, is this clear?
Yeah.
So, but that will not happen if you have. When would that be possible? By the way, it's either going to be possible if you have. Let me copy all of this. So you have 4 ligands of one type and 2 ligands of another type, right? The other place where that could be possible is if you have, so the other one is that you have 3 ligands of one type and 3 ligands of another type. So if you have a 3-3 combination. So in that case, let's say the third one is also water. So that's one of our arrangements. So over here, you can see that, and then there's going to be another arrangement, which is that all the water molecules are on the same side. इसको green में कर लें एक second. So that could be another arrangement. Now, if you look at the arrangements carefully, it's the same exact concept, except for the fact that they don't actually use the opposite side or same side, or I mean, there's a different terminology that's involved. The concept is exactly the same. What you're going to notice is that the water molecules over here are making an angle of 180 degrees on opposite sides. This water molecule is making an angle of 90 degrees with both of them, but at least 2 water molecules are making an angle of 180 degrees. But in this arrangement, if all the water molecules are like adjacent to each other, they're all making an angle of 90 degrees with each other. So there are no 2 water molecules making an angle of 180 degrees. So that's 2 different orientations. So it happens stereoisomers are still formed in almost the same way. You just don't call them cis or trans. You call them something else, which is not part of your syllabus. But you will still be asked to draw this. Okay, so remember, in octahedral complexes, both of these arrangements, whether it's 4 ligands and 2 ligands, or 3 ligands and 3 ligands, both of these arrangements will lead to stereochemistry. Is that clear?
Yes.
Okay, and then there will be certain complexes that in octahedral complexes that will not have stereochemistry. So, for example, if you have 5 ligands of one type and 1 water molecule, then you're not going to have stereochemistry. So in this case, this one is no stereoisomers. Or even if you have all 6 of the same type, then obviously you're not going to have stereoisomers. So it's either 4-2 or 3-3. So either 4-2 or 3-3. So moving on, cis-trans again, there's another type of complex, and there's a specific type of complex which you got to remember, that's cisplatin, and there is transplatin. So shape and stereochemistry. So now the cisplatin is like really important. Why is it important? Because it's an anti-cancer drug. And we'll talk about how cisplatin is basically going to work. Okay, but before that, we'll try and draw what cisplatin is. So cisplatin it's a platinum ion which is surrounded by 2 Cls and 2 ammonias. That's what cisplatin is. It's a complex. Overall, it doesn't really have a charge. So it's not going to have a charge, but it's a complex. So cisplatin has a square planar shape. So there's platinum ion, which is +2, and it's going to be surrounded by Cl-1 ligands. And at the bottom also there's going to be Cl-1 ligands. On one side there's going to be ammonia ligands. So the lone pairs of ammonia will be attracted to the platinum. And overall it's not going to have a charge. So square brackets around it showing that it's a complex and it's got a +2 charge. Okay, and I'm going to redraw this. This is one arrangement. Remember, this is a square planar arrangement, not an octahedral arrangement. So the same molecule can have a different arrangement where what will happen is that the Cls, instead of being opposite to each other, they're actually together. So there's Cl-1 and there is NH3 with its lone pairs. So the only difference is that over here the NH3s were on opposite sides. On the right side, the NH3s are on the same side. And what about the Cls? The Cls were on opposite sides. Over here, the Cls are on the same side. So this one is same side. This one is opposite sides. So these are on opposite sides, whereas those are on the same side. Which is why this is your cisplatin, and the other one, this is my transplatin. And that over there is my cisplatin. Okay, is this clear? Cisplatin and transplatin, they're two drug molecules. You have to remember the shape. I'm going to write down the shape as well. That this is a square planar arrangement. So that's your square planar arrangement, cisplatin and transplatin. And these are like important anti-cancer drug molecules. So you got to remember these complexes as well. Okay, is this clear? And they'll ask you to draw exactly the same thing. Umar, Isaac, Qasim, is this clear?
Yeah.
And now, since we're talking about cisplatin and transplatin, especially cisplatin, it's an anti-cancer drug molecule, so we got to talk about that as well. So this particular, the cisplatin, remember the other arrangement is not that suitable. So I'll just briefly tell you what cancer is, or give you an idea of what cancer is. Cancer is when you have molecules, cells in the human body, which are damaged, and so you've got damaged cells with damaged DNAs. DNA is where all the protein synthesis happens. I mean, that's where the information is stored about which amino acid is going to be attached at what position. So cells with damaged DNAs that replicate rapidly. And what do? They produce tumors. In stage 1, these tumors are very localized. They're in a particular area. So it's pretty easy to find those cells and just kill them by radiation or by injecting chemo. I mean, chemotherapy is basically cisplatin. Then in stage 2, stage 3, and stage 4, these cells which are damaged with damaged DNAs that produce the wrong proteins, they continue to replicate and they spread out. They travel to your lymph nodes. They travel to other parts, to other body parts and other organs, and they start replicating and reproduce uncontrollably, and they produce proteins which are completely useless or damaged. So what you got to do is you got to kill those cells. I mean, that's what the therapy is. So what cisplatin does is that it is basically poison for these cells. It stops them from replicating cancer cells. Now, for a cell to replicate, you've got DNA where all the information is stored. You've got a phosphate group, you've got a sugar group, and you've got a base. This is your nucleotide. And it's like a polymer. The DNA is like a polymer. It's got phosphate group, sugar group, and then base. Now the base is a nitrogen base. It's got lone pairs. So just like ammonia, it can actually form dative bonds. And there are actually two branches going in opposite directions, complementary to each other. This is the ladder-like structure that is stored inside the nucleus of the cell. This is where all the information about protein synthesis is going to exist. So it's a spiral ladder-like structure, and the information that it provides is the position of the amino acid when your body starts to produce proteins. It kind of tells you which amino acid is going to be attached at which position. The order of these bases is what's important. That they basically tell you that this amino acid is going to fit over here and the amino acid is going to fit over here. And the amino acids would start linking up and they would start synthesizing proteins. A part of this chain is copied onto the RNA where it eventually, uh, where the protein synthesis starts happening. Now, when cells replicate, if the DNA arrangement or the sequence of the bases is damaged, that means your cell is damaged, and any protein that it will produce, the cell has forgotten where the amino acid position is going to be. So you've got different amino acids. If a particular amino acid A was supposed to be at this position and a particular amino acid B was supposed to be at this position, if your DNA is damaged and you got the wrong base over here, then obviously the amino acid ordering is not going to be right, and the protein produced is not going to be right. So the cell has to stop replicating. The way it replicates is that it unzips. I mean, the ladder-like structure unzips. And what you get is two half ladders, basically. And the cell splits into two. And then the remaining part of the DNA strand, it gets rebuilt using a portion of the... So it's like you're making ladders. You split the ladder in the middle, and then you try to reconstruct. So that is how cell replication happens, and that's how the information gets copied on to newer cells. And this has been going on in your life forever, since you were born. Your cells are constantly replicating. This information, the ordering of the bases, that's like really, really important. Now, once it's going to get damaged, usually with age, because your cell is replicating so many times, that errors get produced. That is what aging is. That slowly over time, your cell will start to forget how to produce certain proteins. Okay, it's going to start it's entropy. It's going to start losing information. I mean, it's like you're photocopying a page and you're doing it forever. Like, so over time, what will happen is that the photocopies are going to get damaged eventually. And then they'll lead to cancer cells, which are going to be completely useless and then start replicating and they'll start producing tumors. So you got to stop the cell splitting. So what cisplatin does is that it had two CA lines that were strongly bonded to it, datively, and it had two ammonias. But instead, what happens is that the ammonias, they get replaced by the nitrogen bases. And this platinum ion basically ends up forming a complex with the two strands of the DNA. And the cell will not be able to unzip. It will hold the cell together. The cell will not be able to replicate. It will not be able to pass the information. It will not be able to rebuild the DNA strand, split it into two, create more copies of itself. That will stop happening. So that is just a general idea of, I mean, so they're not going to go into detail. They just ask you a very simple question: is how is cisplatin going to prevent DNA replication? And that is that it will bind or form dative bonds or coordinate bonds. So I'm going to write that platinum ions form coordinate bonds with nitrogen bases in DNA, which prevents cell replication. He guess is this so just very basic stuff. Maximum it's going to be a 1 or 2 mark question. That's it. Yeah, but you need to know cisplatin and transplatin. You'll be asked to draw this. Remember, they are square planar molecules. The cisplatin is the one that's the anti-cancer molecule, not the transplatin. Okay. So is this clear? This part? Over
Yes.
clear, Kasan. So we'll leave it at this point, and we're almost done. Okay. Next class we'll do past papers. There's just a few things left. We'll probably finish that in the next class in 10 minutes. And then we'll just do past papers. I've shared the notes for this chapter as well. Okay. चलें ठीक है. Okay, everyone. Allah Hafiz.